Immunity. Introduction. The immune response is how your body recognizes and defends itself against bacteria, viruses, and substances that appear foreign and harmful. Immunology is one of the branch of biology which is the study of for protection from foreign macromolecules or invading organisms and our responses to them. These invaders include viruses, bacteria, protozoa, or even larger parasites. In addition, we develop immune responses against our own proteins and other molecules, and autoimmunity and against our own aberrant cells and tumor immunity. Organisms must find a means of defense against antigens, otherwise, bacteria, fungi, and viruses would replicate out of control and results in the destruction of the body. Therefore, organisms employ many types of defense to stop as happening. Means of defense can be categorized into first, second, and third lines of defense. In this chapter, you will see the overall setup which is running the immune system in our body and how our body triggers responses and employs methods which ensures our healthy survival. First line of defense in our body. In human, the skin is the largest organ of the integumentary system. Skin is regarded as one of the first line of defense because it provides an almost impenetrable biological barrier protecting the internal environment. It protects the body, especially the underlying tissues, against pathogens and excessive water loss. It is also involved in providing insulation, temperature regulation, and sensation. Physical components of the skin defense. Our body's first line of defense is non-specific and includes structures, chemicals, and processes that work to prevent pathogens entering the body. These first line defenders include the skin and mucous membrane of the respiratory, digestive, urinary, and reproductive systems. Skin is comprised of two main layers, epidermis, dermis. The epidermis. The epidermis is composed of multiple layers of tightly packed cells which few pathogens can penetrate their own. In addition to the structural barrier, the natural shedding of dead skin cells removes many attacked microorganisms. Then there are other cells called epidermal dendritic cells that actively patrol the skin to phagocytize, engulf, and digest pathogens. The dermis. The dermis is situated beneath the epidermis and contains protein fibers called collagen. Collagen is a tough fibrous protein which gives skin the strength, pliability to resist abrasions that could introduce microorganisms. Components of the skin's defense. Perspiration. Secreted by the skin sweat glands, perspiration contains salt and enzymes. Few microbes can live in highly saline environments like that of the skin surface. The lysozymes in sweat are type of enzyme that can destroy the cell walls of bacteria. Sebum. Sebum is secreted by skin sebaceous oil glands. The oil helps keep skin pliable and less likely to break or tear, and also lowers the pH of the skin to a more acidic level that inhibits the growth of many types of bacteria. Defense against infection and digestive tract. Proper levels of HCl hydrochloric acid in the stomach are our first line of defense against bacterial and viral infections. All day long, we unavoidably ingest all kinds of bacteria and viruses through the mouth, nose, and food we eat. These germs just cannot survive the trip into the stomach because of acidic environment. The healthy stomach essentially remains sterile. Normally, good bacteria that live lower down in your digestive tract can also move up into the stomach. If it has become more alkaline, proper acid level in the stomach prevent this poor growth from occurring. Cilia and mucus. The most important function of the nose and nasal cavity is to process each breath before it enters the lungs. This filtering process is important for several reasons, considering the amount of air that passes through the respiratory system each day. It is important that the nose filtered out dangerous particles, bacteria, viruses, dust, pollen grains, etc., that could otherwise enter into the lungs, causing damage. Tiny haired cilia and a mucous membrane line the inside of the nose to trap particles before they enter the body. The cilia are capable of small movements and can direct the flow of mucus, a substance secreted by a mucous membrane, removing it from the nasal cavity. Sneezing forces air through the nasal cavity and is also effective at removing particles and mucus. Warm each breath to prevent cold air from damaging sensitive lung tissues. The large amount of surface area and many blood vessels in the nasal cavity help warm each breath quickly as heat transfers from the blood to passing air. Add moisture to each breath to prevent the airways and lungs from becoming dry and damaged. As air passes through the nasal cavity, moisture is transferred from the mucus secreted by the lining of the nasal cavity into the air. Because the nose and nasal cavity are so effective at filtering, warming, and moisturizing each breath, it is generally better to breathe through the nose than the mouth. Though the mouth can allow a person to inhale more quickly, over time, mouth breathing can dry out the delicate tissue in the airways and lungs. Furthermore, oxygen may be easier for the lungs to extract from air that has been filtered, warmed, and humidified through the nose and nasal cavities. The normal lungs, the oxygen from the air is drawn through the nose and mouth, travels down the trachea and to the right and left bronchus and bronchioles, and then finally into millions of alveoli or sacs. From the alveoli, the oxygen is absorbed into the bloodstream and is carried to all parts of the body. To help the breathing process, the lungs have their own natural clearance methods. Your airways produce a thin coat of mucus that catches the dust and bacteria that you breathe in daily. This thin mucus is swept up towards the windpipe by tiny beating hair called cilia, where it can be cupped up or swallowed. Second line of defense. Once pathogens are able to neutralize the responses from the first line of defense, skin and mucous membrane, and are able to penetrate inside the body, they are encountered by second line of defense, which is non-specific because it handles a variety of microbes. Non-specific defense includes macrophages, neutrophils, natural killer cells, the complement system, etc. Rule of macrophages and neutrophils. Monocytes and neutrophils are types of blood cells. As we know that white blood cells are involved in providing immunity to the body. Monocytes are released by the bone marrow. These float in the bloodstream from where they enter tissues and turn into macrophages. Of all blood cells, macrophages are the biggest, most boundary tissues has its own macrophages. For example, alveolar macrophages live in the lungs and keep the lungs clean and disease free by ingesting foreign things like smoke, dust, bacteria, and microbes. Macrophages also swim freely. Once of their job is to clean up dead neutrophils. Macrophages clean up pus as a part of the healing process. Neutrophils are the most common form of white blood cells in the body. Bone marrow produces trillions of white blood cells every day and releases them into the bloodstream. But their lifespan is short, generally less than a day. Once in the bloodstream, neutrophils can move through capillary walls into tissue. Neutrophils are attracted to foreign material, inflammation, and bacteria. If you once a neutrophil finds a foreign particle or a bacteria, it will engulf it, releasing enzymes, hydrogen peroxide, and other chemicals from it granules to kill the bacteria. In a site of serious infection where lots of bacteria have reproduced in the area. Pus will form. Pus is simply dead neutrophils and other cellular debris. Rule of natural killers cells. Natural killer cells are type of lymphocyte, a white blood cell, and a component of innate immune system. Natural killer cells play a major role in the host rejection of both tumors and virally infected cells. Natural killer cells are cytotoxic. Small granules in their cytoplasm contain special proteins such as purpurine and proteases known as granzymes. Upon release in close proximity to a cell selected for killing, purpurine forms pores in the cell membrane of the target cell through which the granzymes and associated molecules can enter, inducing apoptosis. The distinction between apoptosis and cell lysis is important in immunology. Lysing a virus-infected cell would only release the virions, whereas apoptosis leads to destruction of the virus inside. Natural killer cells are activated in response to interferons or macrophage-derived cytokines. Complement system interferons. If pathogenic bacteria get through all the immune system initial defense, the arsenal of the complement system is initiated. The complement system includes proteins and white blood cells, which carry antibodies. The antibodies are macroscopic killing cells that seek out anything they don't recognize as being part of the body. The proteins circulate in an inactive form but in response to the recognition of molecular components of microorganisms. They become sequentially activated working in a cascade where in the binding of one protein promotes the binding of the next protein and the cascade. The white blood cells and the proteins work together to coat any invading bacteria with antigen, which often makes the bacteria incapable of reproducing or binds the bacterium to a toxic chemical to make it break apart. The white blood cells and the proteins work together to coat any invading bacteria with antigen, which often makes the bacteria incapable of reproducing or binds the bacterium to a toxic chemical to make it break apart. The cells in the blood that detect pathogenic bacteria and signal the complement system to that the work are macrophages. The bacteria are passed on to T cells and B cells that are able to recognize and remember how to manufacture the specific antibody to break apart the bacteria. Interferon. Body frequently encounters situations of viruses attacks, although viruses are efficient source of some of the lethal infections, but at the same time, Allah has gifted us with set of proteins which provides an effective shield against viral attacks. The interferons are a group of natural proteins that are produced by human cells in response to viral infection and other stimuli. They have the ability to interfere with viruses that are replicating. There are three main types of interferon: interferons beta and alpha, produced mainly by white blood cells and certain connective tissue cells called fibroblasts. Interferon gamma produced primary activated T cells. Interferon are antiviral agents and can fight tumors. These activities are coordinated by interferons mediated activation of certain immune cells such as macrophages and natural killer cells, and by enhancing cell surface expression of important immune molecules including major histocompatibility complex classes 1 and 2, which display foreign microbial peptides for activation of T cells. Interferon mechanism against viruses. Viral nucleic acids enter cells. Interferon genes of the cell's DNA are turned on. Interferon molecules are produced and released by the cell. The first host cell is killed by the virus. Interferons are received by another cell in response. The second cell makes the antiviral proteins which block viral reproduction. Inflammatory response is one of the non-specific defense. The inflammatory response inflammation occurs when tissues are injured by bacteria, trauma, toxins, heat, or any other cause. These damaged tissue releases chemicals including histamine, bradykinin, and serotonin. These chemicals cause blood vessels to leak fluid into the tissues, causing swelling. This helps isolate the foreign substance from further contact with body tissues. The chemicals are attract white blood cells called phagocytes that eat microorganisms and dead or damaged cells. This process is called phagocytosis. Phagocytes eventually die. Pus is formed from a collection of dead tissues, dead bacteria, and live and dead phagocytes. The main symptoms of the inflammatory response are as follows. The tissues in the area and red are red and warm as a result of large amount of blood reaching the sites. The tissues in the area are swollen against due to the increased amount of blood and proteins that are present. The tissues in the area are swollen against due to increased amount of blood and proteins that are present. The area is painful due to expansion of tissues causing mechanical pressure on nerve cells and also due to presence of pain mediators. Once the inflammatory process has begun, it continues until the infection that caused it has been eradicated. Pyrexia and pyrogens. In case microbes proceeds as establishing a major infection, the body often produces fever, which slows down microbial production and enhances the body's own fighting abilities. Pyrexia, fever, means the elevation of body temperature above the normal range. It may be caused by abnormalities in the brain itself or by toxic substances that affect the temperature regulating centers. Such causes include bacterial or viral infections. Normally, the temperature of the body is regulated almost entirely by nervous feedback mechanism. And almost all of these operate through a temperature regulating center located in hypothalamus at the base of the brain. Neuroreceptors in the skin and spinal cord provide feedback that drives the body to either conserve heat, produce increased quantities of heat, shivering, or increase heat loss, sweating, or panting. Changes in the heat regulating process are constantly undergoing modification that go unnoticed. In other words, heat regulation is not a static process but an ever moving one that is important to normal body function. Substances that may cause the set point of the hypothalamic thermostat to rise are called pyrogens. Many proteins break down products of proteins and certain other substances such as lipopolysaccharide toxins, endotoxins secreted by bacteria can act as pyrogens. It is pyrogens secreted by toxic bacteria are pyrogens released from degenerating tissues of the body. Pyrogens are produced by many microorganisms including bacteria, yeast, and molds. Most potent pyrogens are the endotoxins produced from the cell walls of the gram-negative bacteria. Ways a fever kills microbes. When someone has fever, the body raises the normal body temperature above 37 degrees centigrade to try to kill bacteria or viruses in the body. This temperature is taken orally. Fever is actually the body's natural way of defending itself from invaders like viruses and bacteria because many of them cannot survive in the body due to high temperature caused by fever. High body temperature also signal infection fighting cells of the immune system such as phagocytes, neutrophils, and lymphocytes to defend and help fight off infections. The degree of temperature increase doesn't necessarily correspond to the severity of the illness. The fever response tends to be greater in children than adults. The two functions of fever are to stimulate immune system to create an inhospitable environment for invading organisms. That is to turn up the heat high enough that the invading microbes cannot live. The benefits of fever. More antibodies cells tend to especially attack the exact type of invader that your body is presently suffering from, produced more specific to the agent of disease than any medicines. More white blood cells produced, circulating, mobilizing, and armed to fight off the invading bugs specific to the general category of invader. More interferons produced, which blocks spread of viruses to healthy cells. Boiling up of iron, which bacteria feed on increased temperature, which directly kills microbes. Third line of defense, the specific defense. If a pathogen still manages to cross first and second line of defenses and get into your bloodstream, you are not bitten yet. All cell membranes have protein coats that line the outside of them. Early on, when you were still developing as baby, your body begins learning what cells belong to you and what don't. Now, your developed body has a good idea of whether cells are pathogens or not. This is accomplished by two defense mechanisms, cell mediated and humoral. C lymphocytes are T cells along with macrophages oversee the cell mediated response, while the B lymphocyte of B cells take charge of the humoral response. Monocytes develop from stem cells in the bone marrow. They then go into blood, where they circulate for a while and then migrate into tissues. In the tissue, they further mature into macrophages. Monocytes and macrophages play important roles in the immune defense and inflammation. Lymphocytes are one of the five kinds of white blood cells. Are leukocytes circulating in the blood, although mature lymphocytes all look pretty much alike. They are extraordinarily diverse in their function. The most abundant lymphocytes are B lymphocytes, B cells, T lymphocytes, T cells. B cells are produced in the bone marrow. The precursor of T cells are also produced in the bone marrow but leave the bone marrow and mature in the thymus. Each B cell and T cell is specific or for a particular antigen. Basic types of immunity. This is very important topic for examination. There are two main types of immunity: innate or inborn or natural immunity. The innate immunity system is what we are born with and it is non-specific. All antigens are attacked pretty much equally. It is genetically based and we pass it on to our offspring. It is present from the birth and is inherited from the mother to offspring through the placenta. Acquired or adaptive immunity. It is not present from birth, but it is acquired during one's own life. It is developed by the organism in response to a disease caused by infection of microbes or vaccines. And thus, the protective lymphocytes of body produce antibodies, which not only inactivate the antigens and relieve from an infection disease, but also provide immunity against further attack. It is so as some antibody producing cells persist as memory cells for a long period and produce antibodies immediately after second infection to encounter it. The acquired immunity so developed may be temporary, for example influenza, or permanent, for example measles, mumps, polio, smallpox for a long time. This is main and important point. Types of acquired immunity. Active or natural immunity. It is a long-lasting immunity developed by antibodies produced by an individual's own cells. It is developed in three ways. A. By having the disease, for example, chickenpox, mumps, measles, etc. B. By having a subclinical infection, for example, Sabin vaccine against polio. C. By having killed microorganisms or detoxified toxins, for example, killed stock. Vaccine against polio, tetanus, toxoid against tetanus. Passive or artificial immunity. Passive immunity is borrowed from another source and it lasts for a short time. For example, antibodies and a mother's breast milk provide a baby with temporary immunity to disease the mother has been exposed to. This can protect the baby against infection during the early years of childhood. Everyone's immune system is different. Some people never seem to get infections, whereas others seem to be sick all the time. As people get older, they usually become immune to more germs as the immune system comes into contact with more and more of them. That's why adults and teens tend to get fewer colds than kids. Their bodies have learned to recognize and immediately attack many of the viruses and cause colds. Vaccination aims to develop active acquired immunity. Immunization is the process where our body is made immune or resistant to an infectious disease, typically by the administration of vaccine. Vaccine stimulates the body's own immune system to protect the person against subsequent infection or diseases. The word vaccination or vaccine had been evolved from vacca, which means cowpox, which contains virus for cowpox. The cowpox and smallpox virus is very similar in structure. If the human body is exposed to the cowpox virus through vaccine, the body acquired immunization against smallpox. We can define vaccine as a immunobiological substance which produces specific protection against a given disease. It stimulates the production of protective antibody and other immune mechanisms. Vaccines may be prepared from lab-modified organisms, inactivated or killed organisms, extracted cellular fractions, toxins, or a combination of these preventions of disease as need of the day. The morbidity caused by the disease and rising cost of treating them requires us to focus more on their prevention. Immunization is among the most successful components of preventive medicine. It is effective public health intervention that has had the greatest impact on health of the people. Every year, millions of children around the globe are saved from illness or death because of vaccines. Specific defense mechanisms. Specific defense mechanisms are effective against specific pathogens. There are two main types of specific defense mechanisms involved in immune system. Cell-mediated response, the antibody-mediated immune response. Cell-mediated immune response. The cell-mediated immune response consists of T cells, which originate in the bone marrow but moves to the thymus where their development is completed. T cells are highly specialized cells in the blood and lymph. They fight bacteria, viruses, fungi, protozoans, cancer, etc. within host cells and react against foreign matter such as organ transplants. There are three kinds of T cells: cytotoxic T cells, directly kill invaders; helper T cells, add B cells and other T cells to do their jobs. Suppressor T cells suppress the activities of B and other T cells so they don't overreact in the cellular immune response. Cells of the immune system kill cells of the body that have been infected with a virus or that are cancerous. This response relies on cytotoxic T cells. TC cells contain molecules called purpurine that they release into target cells. The purpurine makes holes in the target cells and thereby kills them. The cellular immune response occurred in two phases. The first, called activation phase, T cells that have the appropriate T cell receptors are activated and triggered to divide repeatedly. In the second, called the effector phase, these activated T cells encounter target cells and kill them. The antibody-mediated immune response. The humoral immune system consists of B cells, which originate in the bone marrow and stay there to develop. B cells can produce antibodies but need exposure to foreign antigens to do so. These antigens are cell surface, oligosaccharides, and proteins. what the cell uses as ID tags. Antibodies are chemically proteins present in blood plasma and lymph. They help in fighting bacteria and viruses in body fluids. All daughter cells of B cells will be able to produce the same antibodies as the mother cell. Antibodies bind to certain parts of an antigen to mark it for destruction by the T cells. The body humoral or antibody-mediated immune response begins in the same manner as the cell-mediated response, but here the macrophages are joined by B cells. The pathogens activate only those B cells with matching receptors. These cells stand ready to enter the battle. Meanwhile, the antigens presenting macrophages activate those helper T cells with matching receptors. These T cells in turn lead the battle front with activated B cells. Triggers by this meeting, the helper release chemicals which make selected B cells to go into rapid reproduction. Some B cells become memory cells ready to respond to a later invasion by same pathogens, but most become antibody producing factories called plasma cells freely circulating in the body. Antibody dock with pathogens thus neutralizes them or marks them for destruction by other weapons in our immune arsenal. Structural model of antibodies. Antibodies are immune system related proteins called immunoglobulins. Each antibody consists of four polypeptides, two heavy chains and two light chains, joined to form a Y-shaped molecule. Amino acid sequence in the tips of the Y varies greatly among different antibodies. Rule of memory cells and immunity. Neither the killer T cells nor B cells just die up after they kill the pathogen. When their job is done, they leave behind memory cells. These memory cells are cells that stay behind and watch for the pathogen. If they find one, they start multiplying to kill it. Memory killer T cells make killer T cells and memory B cells make B cells. This process is so immediate and so explosive that the pathogen is killed off before it has a chance to infect you. Because of that, you are immune to that pathogen. Allergies. Allergies are abnormal reactions to ordinarily harmless substances. The sensitizing substances called allergens may be inhaled, swallowed, or come into contact with the skin. Allergens that most frequently cause problems are pollen, mold spores, house dust mites, animal genders, food, insect bites or stings, plant insects, spores, latex, viruses, bacteria, medications, and environmental conditions such as cold temperature. Although allergies can develop at any age, the risk of developing allergies is genetic. It is related to one's family history of allergy. If neither parent is allergic, the chance for allergy is about 15%. If one parent is allergic, the risk increases to 30%. And if both are allergic, the risk is greater than 60%. Allergens cause the production of immunoglobulin E, IgE, an antibody that all of us have in small amount. Allergic person, however, produce IgE in abnormally quantities. Normally, this antibody is important in protecting us from parasites, but not from other allergens during sensitization period in allergy. IgE is overproduced. It coats contain potentially explosive cells that contain chemicals, including histamine. These chemicals in turn cause inflammation and the typical allergic symptoms. This is how the immune system becomes misguided and cause an allergic reaction when stimulated by an allergen. The most common allergic conditions include hay fever, allergic rhinitis, asthma, allergic eyes, allergic conjunctivitis, allergic eczema, hives, urticaria, and allergic shock. Autoimmune disorders. Autoimmune disorders are diseases that occur when the body produces an inappropriate immune response against its own tissues. Sometimes the immune system will cease to recognize one or more of the body normal constituents as self and will produce autoantibodies, antibodies that attack its own cell tissues and or organs. This causes inflammation and damage and leads to autoimmune disorder. And a few types of autoimmune diseases such as rheumatic fever, a virus or infection with bacteria triggers an immune response and antibodies of T cells attack normal cells because some part of their structure resembles a part of the infecting microorganism. Symptoms of autoimmune disorders vary by the particular disorder, but many include fatigue, dizziness, and low-grade fever. Symptoms can also vary in severity over time. Some of the autoimmune diseases include lupus, a chronic disease marked by muscle and joint pain and inflammation. The abnormal immune response also may involve attacks on kidneys and other organs. Juvenile rheumatoid arthritis, a disease in which the body immune system attacks as though certain body parts such as the joints of the knee, hands, and foot are foreign tissues and attacks them. Scleroderma, a chronic autoimmune disease that can lead to inflammation and damage of the skin, joints, and internal organs. Ankylosing spondylitis, a disease that involves inflammation of spine and joints, causing stiffness and pain. Juvenile dermatomyositis, a disorder marked by inflammation and damage of the skin and muscles. Rule of T cells and B cells and transplant rejection. Transplantation is the act of transferring cell tissues or organs from one side to another. The malfunction of an organ system can be corrected with transplantation of an organ, for example, kidney, liver, heart, lung, or pancreas, from a donor. However, the immune system remains the most formidable barrier to transplantation as a routine medical treatment. The immune system has developed elaborate and effective mechanisms to combat foreign agents. These mechanisms are also involved in the rejection of transplanted organs, which are recognized as foreign by the recipient's immune system. Major histocompatibility complex proteins are involved in the presentation of foreign antigens to T cells, and receptors on the surface of T cells are uniquely suited to recognition of proteins of this type. MHC are highly variable between individuals, and therefore the T cells from the host recognize the foreign MHC with a very high frequency, leading to powerful immune responses that cause rejection of transplanted tissue. Identical twins and clone tissues are MHC matched and are therefore not subject to T cells mediated rejection. The first line of defense is non-specific and is part of the innate immune system. The first line of defense consists of physical barriers, chemical barriers. The second line of defense is greater part. Second line of defense is also part of the non-specific innate immune system and includes non-specific immune cells, chemical mediators, fever, inflammation, phagocytosis. The third line of defense is part of adaptive or acquired immune system. This line of defense provides specific long-term protection against microbes. The third line of defense includes T1, T cells 2, B cells 3 antibodies. Neutrophils are granular, non-specific immune cells that patrol the borders of the body and eliminate microbes by phagocytosis. Monocytes are agranular, non-specific immune cells that circulate in the blood and mature into macrophages when they migrate into tissues. Macrophages are granular, agranular, non-specific immune cells that perform the following function: phagocytosis, professional eaters, sound chemical alarm to alert other immune cells, present information about foreign microbes, antigens to the T cells, which are generally general of the immune army. Phagocytosis as a second line of defense process in which foreign materials, microbes are engulfed and down by neutrophils or macrophages within a digestive compartment known as the lysosome. Inflammation is a non-specific response to tissue injury or infection that walls of damaged or infected tissue. Recruit immune cells to the site of injury and infection. Clear away microbes or damaged cells to tissue. Repair can occur. Fever is an increase in body temperature induced by pyrogens and regulated by the hypothalamus, which decreases bacterial cell growth and increases immune cell activity. Interferons are antiviral proteins released by virally infected cells that help prevent the spread of infection to neighboring cells by degrading key points. DNA, RNA, and blocking production of viral proteins. Complement is a set of immune proteins that aid or complement immune function. Complement proteins form the membrane attack complex, which forms holes on the surface of targeted microbes and lead to their lysis. Vaccine is immunobiological substance which produce specific protection against a given disease. It stimulates the production of protective antibody or other immune mechanism. There are two main types of specific defense mechanism involved in the immune system: cell-mediated immune response and the antibody-mediated immune response. Antibodies are immune system-related proteins called immunoglobulins. Allergens cause the production of immunoglobulin, IgE, and antibody that all of us have in small amounts. Allergic persons, however, produce IgE in abnormally large quantities. When the body produces an inappropriate immune response against its own tissue, it results in the autoimmune disorders.